In past semesters, homework assignment 8 is given students some issues. I want to make sure everyone's on the same page. So I'm going to go over the first page, questions 1 through 5, just to make sure that everyone knows what they're doing. Question number 1 is similar to homework number 7, but I just wanted to reiterate the need to know complementary pairs, complementary pairing in DNA strands. For question number 1, you have the sequence of DNA for the template strand, and I'm asking you to fill in the sequence on the coding strand. I am giving you the 3' and 5' ends to the template strand. So when you fill out the coding strand, you will know that they are on the opposite sides. 5' at the top left and 3' at the top right. And the reason that this is so is because of anti-parallel. When filling in the sequence of the top strand, you will be filling in the complementary pairs that you're seeing at the bottom. So the first nucleotide that we are seeing is thymine. The complementary pair will be adenine. For the next adenine, it will be thymine, guanine, adenine, adenine, guanine, guanine, adenine, thymine, guanine, adenine, 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 and then I fill in the rest for you. You definitely should know how to do this on your own, but I just wanted to make sure everyone knows what I was asking for. For question number 2, you are filling in the messenger RNA sequence of nucleotides when given the DNA template strand. Again, the template strand is 3' on the left, 5' on the right. The messenger RNA will be anti-parallel to this template strand with 5' on the top left and 3' on the top right. When the messenger RNA is created, it will be created in a 5' to 3' direction. So the direction that we are filling in these nucleotides will be the way it is created. And so when filling in the sequence of the messenger RNA, you'll notice that it is very similar to the sequence of the coding strand. The big difference is there are no thymines found in RNA. Instead of thymine, you will be finding uracil. So let's fill out this messenger RNA code just like we did before. We are starting with the thymine, and we will be working our way in this direction. So adenine is the complementary pair to thymine. Uracil is the complementary pair to adenine. Guanine, adenine, adenine, guanine, guanine, adenine, uracil. Adenine, 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 and then we fill in the rest. And as you can see, this code for the messenger RNA is very similar to question number 1, except for instead of thymines, we find uracil. Before looking at question number 3, we want to separate out the messenger RNA, because this will be the molecule that is leaving the nucleus and will go through translation to create the protein. We want to look at the sequences in pairs of 3, the triplets. These are codons. These triplets, these codons, will be translated to one amino acid on the protein. So for question number 3, I am looking for the sequence of amino acids that will be created when translating the sequence of codons found on the messenger RNA. The codons are these triplets that have been separated off. In order to do this translation, you're going to need this graph. And this is the way that we will figure out what amino acid each codon will be translated to. Our first codon is AUG. So how you will decipher this on this chart, on this side is the first base. So our first base is A. So you find the row that corresponds to A, and that is here. The second base is U. So you find the column that corresponds with U, and that is here. Where this column and this row intersect is this cell. Then you take the third base, which is G. You go over to the right-hand side, and you find the row that you're working with. We're working with A, and so U, C, A, G. These are the bases, and we want G, so that means it's going to be at the bottom of the cell that we're looking for. Go back to this cell here at the very bottom. We have AUG, and methionine is our amino acid. So methionine. You will continue to translate each of these codons. Let me step back. The methionine is the start codon. AUG is the start codon. Methionine is the first amino acid. So this will be the first amino acid for all proteins. It later might get cut off, but all proteins, when created, will start with this methionine. All right. So the second codon that we're working with is AAG, and so we'll look for the first base, which is A. So this row here, and then the second base is A. So this column and row intersect here, and then G is our last base, our third base, and that's at the bottom. So then we're looking here. AAG, lysine is our next amino acid. I'll do the third one, which is GAU. G is the first base. A, so that's this column here. And so where G and A intersect is this cell. And then U is at the top. And so you go to this cell and you look for the U, and that is aspartic acid, GAU. So you can go through all of these and figure out for yourself the amino acids that they correspond to. I will go through, make sure what you figure out is what I had figured out up here. Hopefully I am correct in my translating, and so hopefully everything that you find is the same. And now the last one is this stop codon. So UGA, and so U is the first, G, and then A, and so we've got this stop codon here. We'll look a little bit more about what that is, but there is a couple stop codons that are found on this chart. There is only one start codon. So this is the answer to number 3. So when looking at number 4, I wanted you to be able to draw the tRNA molecules and kind of explain the anticodons. And so what is written here is very similar to what we had for question number 3. These are the amino acids that correspond with the codons. But now we want to look at the anticodon. And so basically the anticodon is the base pair that matches up with the codon. So with this first codon, AUG, that's found on the messenger RNA, the anticodon is its complementary pair. So A corresponds with U, UAGC. So UAC is the anticodon of AUG. And why is that important? It's important because it is found on the tRNA molecule. So this right here is my rendition of a tRNA molecule. So the tRNA molecule is going to have lots of different anticodons on there, and the anticodon that matches this codon will attach because they are attracted to one another. Well, for every UAC anticodon tRNA molecule, this methionine amino acid is attached to it. And that is why the methionine is coming to this start codon. The next anticodon is this UUC that corresponds to the AAG, and it is attracted and it's going to bring the lysosine. The lysosine amino acid will be attached to all of these anticodons. When this comes and fits in, you will get this peptide bond that bonds the two amino acids together. So yes, I have this list of amino acids down here, but kind of pretend that they're not there right now. And so as the ribosome is going down this molecule, these tRNA molecules are coming in and bringing the amino acid. So at this moment, we just have these two. When the third one comes in, it brings the aspartic amino acid, and you have the peptide bond. So now you've got this 3 amino acid long chain. This right here, this tRNA molecule will fall off and will just leave this amino acid. So as the ribosome is going down, you will only have just 2 tRNA molecules in place. As the ribosome moves down, they start to fall off, but you leave the amino acid and you leave this chain. So going down, you can see how the anticodons correspond to the codons and bring these amino acids to play. But when you get to this UGA is this codon. The anticodon is actually, there is no tRNA molecule that will be bringing in an amino acid. Instead, you will have this large protein matching up, and it is a release factor protein. So it'll be matching up and it will fit in. And this kind of breaks the chain of the amino acid building, and it will release the entire amino acid that was just created. It will release the protein. If this ribosome is free floating in the cytoplasm, then it will release it into the cytoplasm. If it is attached to the rough ER, the rough endoplasmic reticulum, then this protein will go inside the endoplasmic reticulum and kind of be shaped and formed, and things will be added. So for question number 4 and 5, for question number 5, and then later question number 6, which you will do on your own, there is a mutation. A point mutation takes place. And so the idea here is to see how this amino acid chain, how this protein may or may not be changed compared to the point mutations that took place. So if you read it, a mutation occurred on the 4th base in the template side. And so the 4th base is a thymine, and that is on the template. And thymine has been substituted with guanine. So this template, the guanine has been substituted in instead of the thymine. Now up top, we've got this messenger RNA molecule that corresponds to that original template that we had. But when this messenger RNA molecule is created from the template that we now have, that code will not be the same. That adenine, which used to bond to the thymine, now will be a cytosine, because the cytosine will be put in there to correspond with that guanine. So now this is our new messenger RNA molecule. So now we need to translate the new messenger RNA and figure out the amino acid chain that will be created. So I have this mutation shown here, but this list of amino acids comes from the original. So this lysine is coming from the original messenger RNA before the mutation. So let's take that away. Now, the mutation, so that was lysine. Let's figure out what amino acid this mutation will create. CAG is our new codon. C, A, so it'll be here, and G, which is found at the bottom. So we have glutamine. So CAG equals glutamine. So instead of lysine, which was our last amino acid, we've now substituted it with glutamine. Now, this is one single change in the amino acid chain. How much of a difference will that make in the shape of the protein? Unknown. But it could be a dramatic difference in the shape. It could affect the protein significantly. The protein might not do what it needs to do at all, and could have a big change there. So those were the answers for questions 1 through 5, kind of giving you a start to what I'm looking for. Please now go through 6 through 14 and answer them on your own. Be aware, questions 11 through 14 are talking about mutations to the coding strand of the DNA. You will have to figure out what that mutation will translate to the new template sequence of DNA, new DNA template sequence. In turn, that template sequence will then create a new messenger RNA sequence, and then finally you'll give me the new amino acid. If you have any questions, please email me and let me know, and I will do my best to help you out. Hopefully this is a good start, and will put you on a good path.